Welcome to Sir Einstein Academy. We are going to start rapid sessions of subject biology 12th CET. But first we will going to start with 11th topic. First topic is respiration and energy transfer. First of all, we need to understand the term phosphorylation. What is phosphorylation? Formation of ATP is called as phosphorylation, which occurs in three different ways. First is photophosphorylation, second is substrate level phosphorylation, and third one is oxidative phosphorylation. What is first one? Photophosphorylation. The formation of ATP which takes place during photosynthesis is called as photophosphorylation. Second, substrate level phosphorylation. Direct phosphorylation of ATP by transfer of phosphate from the suitable substrate is called as substrate level phosphorylation. Site of the substrate level phosphorylation is cytoplasm of cell and matrix of mitochondria. Third one is oxidative phosphorylation. What is oxidative phosphorylation? Where we need to invest ATP, that is phosphorylation of ADP at the cost of energy is called as oxidative phosphorylation. Site is inner mitochondrial membrane. Next is respiration. What is respiration? Respiration is a catabolic process. What is catabolism? Breakdown of complex molecule into simple molecule is called as catabolism. So respiration is a catabolic process in which complex molecule are oxidized into simple components. Anaerobic respiration. There are two types of respiration. One is anaerobic, second is aerobic respiration. In anaerobic respiration, there is no involvement of oxygen. That is, fermentation is a type of anaerobic respiration. Glycolysis. What is glycolysis? If we break the word, we will understand glycos is sugar, lysis is breakdown. So the breakdown of sugar is called as glycolysis, which takes place in 10 steps. Glucose is converted into 2 molecules of pyruvic acid by the 10-step reactions. Cytoplasm of the cell. Glycolysis takes place in the cytoplasm of cell. Now, there are 2 phases of glycolysis. First 5 phases are called as preparatory phase, where we need to invest ATP. Last 5 phases are called as, or last 5 steps are called as payoff phase, in which we gain ATP. So, if we begin with glycolysis, first of all, remember all the students have to study all the enzymes of these steps which are given in your textbook. First, we get glucose. Glucose is converted into glucose 6-phosphate. After that, it is converted to fructose 6-phosphate. Then fructose is converted to fructose 1,6-diphosphate. All these 4 molecules are of 6 carbon each. Now, here what happens, fructose 1,6-diphosphate is converted into 2 molecules of 3 carbon each. One molecule is glyceraldehyde 3-phosphate, second is dihydroxyacetone phosphate. These two are enantiomers of each other, that is, they can be interconvertible. If we take glyceraldehyde 3-phosphate, 2 molecules, then it is converted to 1,3-bis-phosphoglycerate, it is converted to 3-phosphoglycerate, after that 2-phosphoglycerate, 2-phosphoenol pyruvate, and lastly we get pyruvic acid. All these molecules from here are of 3 carbon each, and here we get 2 molecules of 3 carbon each. Last is the pyruvic acid, which there are 2 molecules. So this overall glycolysis is divided into all these 10 steps. Here in the first 5 steps, that is in the first and the third step, we need to invest ATP. So in all total we invested 2 ATPs. How many ATPs we invested? 2 ATPs. Lastly, in the last 5 steps, we get 2 NADH plus H plus. We know that 1 NADH plus H plus is equal to 3 ATPs. 2 NADH plus H plus is equal to 6 ATPs. Here, plus these 2 ATPs, and lastly, at the 10th step, we are getting more 2 ATPs. So, this 6 plus 2, 8, and this 2, 10. So, total ATPs we are gaining is 10. Net gain of the ATP in glycolysis is 10 minus 2, that is equal to 8 ATPs. Next point we are going to see is fate of pyruvic acid. The pyruvic acid which is formed in glycolysis, it will either undergo lactic acid fermentation, or it will undergo alcoholic fermentation, or it will undergo complete oxidation. First we will see lactic acid fermentation. What is lactic acid fermentation? When after vigorous exercise, when our body, when our muscles get drained, the pyruvic acid is converted into lactic acid. When we take some rest, after that lactic acid is converted back into pyruvic acid. Second is alcoholic fermentation, which takes place in yeast. Pyruvic acid is converted into acetaldehyde. Along with that, CO2 is evolved, and that acetaldehyde is converted into ethanol. This is called as alcoholic fermentation, which takes place in yeast. The third fate of pyruvic acid is aerobic respiration, where complete oxidation of glucose takes place. Final electron acceptor is the molecular oxygen. Now we will see what happens. This step is also called as the connecting link between glycolysis and Krebs cycle. Here pyruvic acid converts into acetyl-CoA where carbon dioxide is evolved. This series of reactions takes place with the help of pyruvate dehydrogenase complex. This reaction is called as intermediate between glycolysis and Krebs cycle. Enzyme involved is pyruvate dehydrogenase complex. Here CoA SH adds and NAD is converted into NADH plus H plus. The site of this reaction is different in prokaryotes and in eukaryotes. In prokaryotes it takes place in cytoplasm and in eukaryotes it takes place in mitochondria. The type of reaction is asked usually. This type of reaction is called as oxidative decarboxylation reaction. It is also asked that connecting link between which two cycles. This step is connecting link between glycolysis and Krebs cycle. Point is Krebs cycle, which is also called as tricarboxylic acid cycle or TCA cycle. Another name is citric acid cycle. The site of Krebs cycle is mitochondria of the cell. It is also called as amphibolic pathway. Why? Because in this Krebs cycle, we can see both catabolic reaction also and anabolic reaction also. What is anabolic reaction? In which simple molecules are converted into complex form by investing the ATP. Oxidation of 1 NADH plus H plus gives 3 molecules of ATP and oxidation of 1 FADH2 gives 2 molecules of ATP. Now, when we say actual Krebs cycle, the intermediate reaction that we have seen earlier, that is pyruvic acid, is converted into acetyl-CoA. After that, acetyl-CoA along with oxaloacetic acid enters into the cycle and it forms the first compound, which is called as citrate. After that we get cis-aconitate. After that isocitrate. After that oxalosuccinic acid. Now in this step of isocitrate to oxalosuccinic acid, here NADH plus H plus is formed. After that alpha ketoglutarate to succinyl-CoA. Now here also in the 6th step also we get NADH plus H plus. So in the 4th step and in 6th step we are getting NADH plus H plus. Next step is succinyl-CoA converts into succinate where there is formation of 1 GTP and 1 GTP is equal to 1 ATP. Succinate converts into fumarate where FAD is converted into FADH2. Fumarate is converted into malate and lastly malate is converted into oxaloacetate. Here also we are getting 1 molecule of NADH plus H plus. So total in all in this Krebs cycle, 1 molecule of pyruvic acid when it is converted into oxaloacetate, we are getting 3 molecules of NADH plus H plus, we are getting 1 molecule of GTP and we are getting 1 molecule of FADH2. Here we know that intermediate step we are getting 2 molecules of pyruvic acid. So for every glucose we have 2 molecules of pyruvic acid, which means we have total 6 molecules of NADH plus H plus, 2 molecules of GTP and we have 2 molecules of FADH2. It is electron transport system in which electrons are transported between these 5 complexes. First of all we will see the names of 5 complexes. Complex 1 is NADH dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is cytochrome BC1 complex. Complex 4 is cytochrome C oxidase, which has A and A3. Last complex is F0F1 ATP synthase, which synthesizes ATP. Now we are going to see what happened in these complexes and lastly we will see to whom they transfer their electrons. First complex NADH dehydrogenase. Here NADH plus H plus is oxidized. This complex transfer its electron to ubiquinone, that is CoQ. This ubiquinone when reduced it is called as ubiquinol. Second complex succinate dehydrogenase where FADH2 is oxidized, they also transfer their electrons to ubiquinone. These two complexes transfer their electron to ubiquinone. Now ubiquinone transfer its electron in the third complex. The third complex is cytochrome BC1 complex where ubiquinol is oxidized. Ubiquinol which gets the electron from these two complexes. Now here this ubiquinol is oxidized in the complex 3. So complex 3 cytochrome BC1 complex oxidizes ubiquinol and transfer its electron to cytochrome C. The fourth complex is cytochrome C oxidase. The name itself shows that here cytochrome C is oxidized and the electrons are transferred to the last electron acceptor, which is molecular oxygen. And so this step is called as terminal oxidation. Here molecular oxygen is the last electron acceptor. So if the question is asked who is the electron acceptor or the last electron acceptor in electron transport system, it is molecular oxygen. So this step is called as terminal oxidation. All these complexes transfer an electron to their first complex transfers to ubiquinone, second transfers to ubiquinone, third complex transfer to cytochrome C. After that, fourth complex transfer its electron to molecular oxygen. Last complex 5, that is F0F1 ATP synthase. What happens actually in this complex is protons from the outer chamber of the mitochondria are transferred to the inner chamber of mitochondria. The F0 play important role in transformation of protons and F1 is the actual site of ATP synthesis. When the protons are transferring from outer chamber to inner chamber, the conformational changes takes place in F1 where it become active and it start producing ATP. After that, it leads to the synthesis of ATP and this step is called as oxidative phosphorylation, as we have seen in the beginning of the lecture that what is oxidative phosphorylation? It is the synthesis of ATP. And now here this step is called as chemoosmosis. Next point is balance sheet of ATP. For 1 glucose, we know that 1 glucose means 2 molecules of pyruvic acid. Here we will see each and every reaction, how many ATP are formed, how many ATP are consumed, how many NADH plus are formed, how many FADH2 are formed. In glycolysis, 2 ATP molecules are consumed. In pyruvate, that is intermediate step, that is pyruvate to acetyl CoA, no ATP is consumed. Even in Krebs cycle, no ATP is consumed. Next, ATP produced. In glycolysis, we get 4 ATP. In pyruvate to acetyl CoA, that is an intermediate step, no ATP is produced. In Krebs cycle, 2 ATP molecules are produced. Next, NADH plus H plus. In glycolysis, we get total 2 NADH plus H plus and we know that 1 NADH plus H plus is equal to 3 ATP. So, we have multiplied it by 3, total 6 ATPs. Same intermediate step 6 ATPs and the last Krebs cycle step 18 ATPs, because there are 6 molecules of NADH plus H plus for 2 molecule of pyruvic acid, means for 1 molecule of glucose, total 18 ATP. FADH2 we directly get in Krebs cycle, how many? 1 for each pyruvic acid, that is 2 for 1 glucose and total 2 into 2 4 ATP. Lastly total 10, 6, 24. The net gain or the balance sheet if we see, we get total 40 ATPs in which 2 ATPs are consumed in glycolysis. So, the net gain is 38 ATP. Lastly, we are going to see respiratory quotient. What is respiratory quotient? The volume of CO2 released upon the volume of oxygen consumed is called as respiratory quotient. The respiratory quotient of all these 3 are asked. Carbohydrate respiratory quotient is 1, for fats respiratory quotient is 0.7 and for proteins respiratory quotient is 0.9. During anaerobic respiration where oxygen is not involved, respiratory quotient is infinite. Thank you. This is all about respiration and energy transfer.